Hi, everybody. Welcome to chapter 16. This is going to be the lymphatic and immune system. So, like always, we'll start with the different components. So, the lymphatic system is going to be composed of lymph vessels, lymph nodes, which sometimes you guys might notice if you get sick, the lymph nodes start to get a little swollen and sore, commonly like in your neck area. The spleen is also part of your lymphatic system, and then the thymus. The function of the lymphatic system is going to be to return excess tissue fluid and then also help with the immunity. So here are going to be the like visual components of it, right? So you have lymphatic capillaries, vessels, trunks, and ducts. And these are just different sizes of the tubes that are going to carry lymph to and from different areas of your body. Lymphocytes are going to be the cells of the lymphatic system. And then there's different types of lymphoid tissue. In the nodules, you have what we call MALT, which I'll explain a little bit later on. It's like a mucosal associated lymphatic tissue. You have the tonsils, you have Peyer's patches, and then the appendix. And then the lymphoid organs are going to be, again, the nodes. The nodes are going to be anywhere you see the little bulges in there. The thymus, which sits anterior to the heart, the spleen, which sits on the left side of the body, and then also bone marrow. So one more time with a little bit more depth, the functions. So functions here are going to be maintenance and distribution of lymphocytes, and then other immune system components. Also, they help with lymphopoiesis, which is the creation of the lymphocytes. Also, returning excess tissue fluid to the cardiovascular system. And that's going to help maintain your blood volume in your body. Next is absorption of lipoproteins from the GI tract. The reason why your lymph is going to absorb the lipoproteins is because these are very large and they can't go through blood. So lymph vessels are going to be a little bit larger. So that's how these lipoproteins get transported. And then lastly, you have transport of immune system components like the white blood cells, and then other fluid and nutrient transport. So lymph formation, when you look at the capillary beds here, right, these are arteries, these are veins, these are the capillary beds, you'll notice that the lymph runs directly through them. So they kind of just run in parallel with each other. Like you'll see the green is a lymphatic duct that runs through the capillary bed. So the capillary beds are to maintain osmotic and blood pressure components. The result is going to be fluid into the tissues. The retrieval mechanism then is fluid entering blind lymphatic capillaries. Blind means they're only open on one side. So you guys can see that they're closed on one side over here too. And then the lymph content is going to be interstitial fluid, lymphocytes, and then macrophages, which again, macrophages munch. They're going to phagocytize anything old, dead, or dying. So the lymphatic capillaries in here, again, they have blind ends, and the blind ends, again, mean that they're closed on one side, open on the other. Their walls are very thin, and they have an incomplete basal lamina. The endothelial cells, they tend to overlap, and the overlap there is going to be helpful to create one-way valves for the extracellular fluid to keep going in one direction. And then it also helps with anchoring the tubes in place. And then lastly, the lymphatic capillaries are going to be a trap for fluid to grab things and keep the fluid inside. So when you look at lymphatic vessels, this is a good image of a cross section. We talked about arteries and veins last time. You have a small lumen for the artery, but a thick muscular wall. For the vein, you have a large lumen and a thin muscular wall. When you look at a lymphatic vessel, you'll notice that it's very irregularly shaped, but the wall itself, in terms of its composition, is going to be similar to that of a vein. It does have valves inside of it, which help, like here, to create one-way flow, right? So you'll have flow go past there, and then just like in the veins, you'll have one-way valves that are going to close off to continually force the fluid in one direction. The ducts are larger here, and they have smooth muscle in their walls. And then very much like your vessels, the skeletal muscle pumps are going to help move the lymph. And again, very similar to your venous system for return. So flow of lymph, you have fluid and proteins that are going to be escaping from the vascular capillaries. Those are going to be collected by lymphatic capillaries. and then taken to vessels, trunks, and ducts. And these are capillaries to vessels, vessels to trunks, and trunks to ducts. Basically, you get bigger and bigger and bigger as you go along. The respiratory and muscular pumps are going to promote flow into the lymphatic fluid, and then the lymphatic ducts are going to empty into the subclavian veins, like you can see here or here. From there, you're going to go into the thoracic duct. So the subclavian veins have been blown up, but you can see how they're going to be draining into the thoracic duct there. And the thoracic duct has a component that's called the cisterna chili. It's going to be at the base of the thoracic duct, and it kind of just bulges out a little bit. So it's going to sit right to the right and left side of the body below the diaphragm. Then you have the left side that's of the body that's going to be above the diaphragm, so like here. And then they're going to empty into the subclavian veins, so it goes up and then into there. The right lymphatic duct is going to be on the right side of the body above the diaphragm, and then it's going to empty into the right subclavian vein. So lymphatic tissue and organs, the primary lymphatic organs are going to be the red bone marrow, the thymus, and then the sites of stem cell origin. The secondary lymphatic organs are going to be the lymph nodes, spleen, and tissues, as well as the immune system responses. So going one by one, primary lymphatic organs, red bone marrow. So you'll see this is the outer surface of bone, and then you have yellow marrow on the inside. That red marrow in between there is going to be that primary site for lymphatic organs. And then also you have the thymus, and again, that lays anterior to the heart. It's that gland right there. The thymus is really unique. It anatomically has a capsule with two lobes, and then a septa, which is basically the lobules, right? The like left and right side. The outer portion has a cortex where you find thymocytes, which are the cells of the thymus. Thymosin and thymopoietin are hormones that are going to send signals to help develop thymocytes and help mature them into T cells. Now, your T cells are going to help your body by fighting off foreign pathogens, or you would just say like a viral or bacterial infection. And then the medulla of the thymus is going to be where the macrophages are located, and then also the dendritic cells. The secondary lymphatic tissues, so the lymphoid nodules are going to be in like the respiratory area, digestive, urinary, and reproductive tracts. These are going to be densely packed lymphocytes that tend to clump together and fill up when you are sick, because you'll have more white blood cells in your body when you're sick and fighting an infection, which is why it becomes sort of tender or sore in those areas. And then the extracellular connective tissue matrix is also present in the lymphoid nodules, and these are where your germinal centers are present. Also, you have MALT, which again is mucosa-associated lymphatic tissue, tonsils, Peyer's patches, and the appendix. So starting with the lymphoid tissues with MALT, these are going to be densely packed lymphocytes in germinal centers. So you'll see them very, very tightly compact here or here or here. Tonsils, you have a couple different sets of them. So you have pharyngeal tonsils, which are going to be these ones right here. They're also called adenoids. Those are going to be in the posterior superior wall of the nasopharynx, which basically means at the back of the nose. Then you have the palatine. The palatine tonsil would be close to the hard palate or the roof of your mouth. And then you have the lingual. The lingual are going to be underneath the tongue. Okay, so the spleen. So this is the spleen right here. Sits inferior to the stomach. The capsule portion is the outside part for protection. And then you have trabecular arteries or veins, and veins. So the veins are going to kind of make this trabecular network, which looks really, really web-like. Inside of the spleen, you have two different areas. You'll have a red pulp and a white pulp. So you guys can see here, this is the white pulp, and then outside here is the red pulp. The red pulp is going to be a venous sinus that has the macrophages, which are going to filter out old red blood cells, platelets, or microorganisms. And again, macrophages munch, they're going to essentially eat them up. The red pulp is also where you have a reserve of red blood cells, which is where you get the color from. And then the white pulp inside of there is the lymphocyte storage in the splenic nodules. So that's where all your lymphocytes sit until you need them when you become sick. Okay, so here's a lymphoid nodule blown up. It has a capsule, which is the outer part, the black line. Then you have like the trabecular network and afferent capillaries that lead in. Then you have what's called the hilus. The hilus is this region where the efferent capillaries are going to lead out. You have a subscapular sinus, which is going to be underneath the capsule. That yellow area all the way around is the subscapular sinus, which has reticular fibers, macrophages, and dendritic cells. That's how you typically identify as the spleen because it has the reticular fibers, which stain very dark histologically. The outer cortex is going to have B lymphocytes and germinal centers. The deep cortex is going to have the paracortical sinus, which is where T cells are going to develop. And then the medulla, which is the core, is going to have macrophages and medullary cords inside. So again, they're pointing to like the medulla in this area, the medullary sinus. You have the trabeculae running in, and the cortical sinus, and we already talked about the efferent vessel. So again, distribution of the lymph nodes. You're going to have some in the cervical region, so around the neck, in the armpit or axillary region, in the thoracic region, which is the thorax, the abdominal region, intestinal region, uh, so it's called mesenterial. The mesenteries are going to be what help keep all the organs in the abdominal pelvic cavity in place because when you do things like go upside down, right? If you're doing like a handstand or whatever, your organs do not fall with gravity, right? The organs wouldn't fall into the chest. So, like what I'm saying is your stomach and your small intestine, your large intestine don't fall into the chest, and that's due to the mesentery present. The mesentery is going to basically adhere all of those organs to the cavity walls. And in the mesentery, you also have lymph nodes, which are called the mesenterial lymph nodes. Then you also have the inguinal region here where you have lymph nodes, and then the popliteal, which is back of the knee. So this is a good image for you guys to see. This one is the crested papillae on the cheek, and then the cervical lymph nodes are bulging here. Here in this case, you're looking at thoracic. Here is popliteal, and then here is axillary. Okay, so that takes us out of the lymphatic system and into the immune system. So the immune system is going to function for your body's benefit to help keep out foreign substances, to keep homeostasis in the body, which is health. So textbook definition, immunity is the ability of the body to protect itself from foreign substances and cells, including disease-causing agents. So there's two general types of immunity. One is called non-specific, which means innate. Innate basically means you're born with it. So the immune system that you're born with. And then the second one is specific or adaptive. That's going to be the one that you build over time due to like exposure of different things. And this can be either active or passive. So you would also call that natural and artificial. Natural could be passive or active, which is maternal or infection. And then artificial is like antibody transfer or active immunizations. Okay, so a couple key terms before we move on. They sometimes look hard to pronounce or might be hard to pronounce, but they're actually not that bad. So the first one is an antigen. An antigen is a foreign substance that stimulates the immune system to react. Usually what's going to happen is a protein or polysaccharide will respond. An antibody or immunoglobulin is going to be a globular plasma protein produced in response to the antigen, and it's going to be specific to that particular antigen. So it's the thing that's going to help kill off the foreign substance. When you put both of them together, we'll call it the antigen antibody complex. A complex is just where you put multiple things together. The complement system is going to be a procedure in which proteins in the plasma that are non-specific defense get paired with an antigen antibody reaction in order to kill off foreign substances. And then last terminology here is the major histocompatibility complex. We actually have two of them, so we'll call them the MHC1 and the MHC2. All cells have an MHC1 except for red blood cells. And then antigen presenting cells are going to have an MHC2. So hold on to those terms while we work through the rest of this. Okay, so that takes us into this, and it's telling you these are types of non-specific immunity in the body. These are types of specific immunity, and these are types of induced immunity. So if you go back, non-specific means innate, which means you were born with it. So barriers to entry, that's your skin. Inflammatory reaction. If a bug bites you, you're going to have some inflammation in the area. Phagocytes, right? Those are macrophages. Natural killer cells. Those are literally a type of cell in your body that help kill off foreign things. Protective proteins and fevering. So fevering actually causes your body to create an inhospitable environment for the foreign substances, which basically means they don't want to live there because it's too hot. So it also helps to kill off in that way. Specific immunity is going to be antibody mediated immunity or cell mediated immunity. And then again, induced is either going to be active or passive. So we'll start with non-specific and barriers. Again, this is something like your skin or your mucous membranes. And again, remember the mucous membranes are going to line the areas of the body where you have the internal body open up to the external environment. PCCE, right? That's going to line the trachea. And the idea in lining the trachea is that it's going to help get rid of like mucus when the mucus in your trachea or throat catches things like dust or dirt or bacteria or viruses. Tears, saliva, and oil gland secretions, they all release lysozymes. Lysozymes are going to be an enzyme that has antimicrobial agents inside of it in order to kill things before they get too far into your body. And those are going to be commonly found in places that open the external environment to the internal body. So, like the eyes, where tears come out, the saliva, where mouth is, and oil glands, where you obviously open up oil glands in your skin. Other barriers are like stomach acid or vaginal acid. Basically, the really acidic environments tend to kill off a lot of substances because it's so far outside of their optimal pH, they die. And the last parts of vomiting, urination, and defecation, that those are ways to get rid of like excrete substances out of your body that shouldn't be there. And so like maybe you get food poisoning and your body does its best to get rid of all the bacteria in the food poisoning, but unfortunately it's too much for it to take care of on its own. So you will literally get rid of it by throwing it up. These are all going to be called your first lines of defense, right? You don't have to think about them, you're just born with them. Next is an inflammatory reaction. So there's 4 different signs that you're having an inflammatory response: redness at the site, heat, swelling, and pain. In this case, mast cells are going to release a chemical that's called histamine. Histamine is going to cause your blood vessels to vasodilate, and it's going to increase the amount of red blood cells going to that area, which will cause more redness and more heat. Also, histamine will allow the capillaries to be a little bit quote unquote leaky, which just means that they're starting to swell and allow things through. And then the pressure on the free nerve endings will lead to the pain. Phagocytes is the next one. So there's 4 different stages of phagocytosis, and this is where you engulf a substance and then you break it down and get rid of its debris. So the first part is chemotaxis. This is literally it's a chemical signal that phagocytes are going to be attracted to that are given off by damaged tissue, complement proteins, or microbial products. So basically, it's letting the phagocyte know, like, this thing over here, it's foreign and you should come get it. And again, that's due to the chemical that's released by a process called chemotaxis. The second part is adherence. This is where you have the attachment of the plasma membrane of the phagocyte to the foreign thing. And in this case, they're calling it bacterium. Number 3 is ingestion. It means you're literally going to pull, right? Endocytosis, you're going to pull in the bacteria and you're going to just put it into this little vesicle for transport. So when you pull it in, it's literally called engulfment by pseudopods. These are fake little cells that kind of walk around and they grab it in. When they adhere and ingestion occurs, this is called opsonization. It's just a fancy word for pulling it in. Then you have digestion and killing, which basically means that you're adding the lysozymes to the bacterium. And in a closed vesicle, what's going to happen is the lysozymes are going to digest the bacteria. And then from there, you're going to exocytose the waste or the debris. And the 3 different types of white blood cells that help here are neutrophils, monocytes, and macrophages. I could go a little bit further and say that neutrophils are going to be the cells that identify the bacteria as foreign and try to help kill them off. Monocytes are going to differentiate into macrophages, and macrophages munch or engulf the cell and do the phagocytosis. Next is natural killer cells. Natural killer cells are going to be cells that lyse other cells, which means they cause them to burst with abnormal MHC1 proteins. They're going to help recognize self from non-self cells and protect, against viruses and tumor cells. The next part here is protective proteins. So these come in the form of a pathway called the complement system. Here you have proteins that are going to be produced by the liver. And the end goal here is they're going to create what we'd call an MAC. The MAC is a membrane attack complex. So if you're looking at this green bubble here, we'll call that the invading pathogen. So that's the thing that your body wants to kill. You're going to go through this process of putting these different proteins together in order to get a protein complex, which is going to allow essentially a hole to be created in the surface or the cell membrane of the bacteria. When you create a hole in the bacteria, what's going to happen is water's going to rush in the cell, the bacteria, because it's a cell, the bacterial cell is going to swell and swell and swell until it bursts. Because structure relates to function, when it bursts, it loses its function, which means it dies. So that's a good thing. That's the idea, right? You want to kill this thing. So the complement system is going to respond to an antibody antigen complex, but it is not specific to the antigen. So it could do it with really any antibody. Next, we have protective proteins. So protective proteins are sometimes called interferons. These are going to be proteins that act in a paracrine hormone fashion, meaning around the cells next to them, and they cause the nearby unaffected cells to form translation inhibitory proteins and then RNA ACEs, which are going to slow down the spread of the virus and activate macrophages and natural killer cells. They're actually really beneficial because all of these contribute to the illness in the body, right? The viruses are harmful because they can multiply and mutate very fast. But if we can slow down the spread of the virus, that's great. Also, they're going to activate macrophages, which are going to come munch up all of the debris, and then the natural killer cells, which will also attack the pathogen. Next is a fever. So the fever is going to be macrophages releasing cytokines. Those cytokines you can also call pyrogens. Pyrogens are going to be released in response to pathogenic molecules, and they're going to stimulate or call upon the anterior hypothalamus, and they're going to cause an abnormally high body temperature, which we would call a fever. So when a fever is produced, you have an increase in environmental temperature, which is above the optimal point for lots of different microorganisms. And because it's so high outside of their normal temperature or their optimal temperature, their cells are going to go into shock, which are then going to kill them. And then also production of heat shock proteins. It's going to be recognized by the T lymphocytes, which are going to come help kill the foreign invaders. And then lastly, you'll have an increase in enzymatic reactions and metabolism. Fevers can be beneficial for us unless they're too high for too long. Obviously, if you have a very high fever for very long, your cells are going to start to shut down because your own body cells are going to be outside of their optimal environment for too long as well. Okay, so specific resistance, when talking about immunity, immunity is the ability of the body to defend itself against specific invading agents. So we have antigens here, like bacteria, toxins, viruses, pollen, etc., right? These are all things that are foreign to your body. Specificity is the ability of the body to recognize self from non-self cells. That's a great thing because when your body recognizes, oh, this is of my own body and I shouldn't kill it, it allows it to continue to function. When your body recognizes, oh, this is not supposed to be here, it will kill it. And then memory cells are going to be important because once you're exposed to a certain pathogen, the second time you're exposed to the same pathogen, your memory cells activate and they help the response occur in a stronger fashion, but also quicker. So it's a twofold thing, and it's beneficial on both sides. And then also you're going to have the immunity that's going to be mediated by lymphocytes. So types of immune response, again, there's going to be antibody mediated or cell mediated. So antibody mediated is also called humoral. This is where you have the destruction of antigens due to the presence of the antibodies. There's going to be a defense against antigens dissolved in body fluids and extracellular pathogens or bacteria that multiply in the body fluids, but these rarely ever enter the body cells. Now, cell-mediated immunity is where you have destruction of the antigens by T cells. So this is where you have them effective against intracellular pathogens, so like fungi, parasites, viruses, sometimes cancerous cells, and then foreign tissue transplants. So anything that your body is like, this is not of my own, I need to start killing it. Your cell-mediated immunity is always going to involve cells attacking cells, and again, T cells attacking other foreign cells. The pathogens are usually going to provoke both types of immune response. Your body's idea when it gets something foreign is it attacks very hard and as fast as possible. And even with that, sometimes you still see symptoms of the illness. So the reality is when something foreign enters your body, there's lots of different mechanisms in place to try to kill it off as fast as possible because there's not just one cell that's coming in that's making that's foreign, right? There's hundreds or sometimes thousands of foreign cells that your body has to kill. So it does all of these things at one time. Okay, so maturation of the T and B cells. So B cells are going to help with antibody-mediated responses. Really, what they're going to do is they don't attack the cell themselves, the foreign cell themselves. They create plasma cells that are going to form the antibodies. The antibodies then are going to attack the foreign cell. So they're going to be effective against bacterias. T cells are going to, again, mature in the thymus. These are going to be in cell-mediated responses. They're going to help by differentiate into killer T cells that are going to directly attack the antigen or helper T cells that are going to stimulate the production of more T and B cells. And these are going to be effective against fungus, a virus, a parasite, cancer, and then also tissue transplants. Okay, so with antibody-mediated immunity, you have defense against the viruses and bacteria. The B lymphocytes are going to mature in bone marrow and then mature into plasma cells, which produce antibodies. You'll have something that's called the clonal selection theory, which states that the specific antigen activates a specific B cell, which then is going to undergo clonal expansion, which basically what that means is after the infection is gone, you're going to have some of those cells become memory cells and turn into your body's long-term immunity, which is good. And then the rest of them are going to undergo apoptosis, which is spontaneous cell death. So this is what that looks like, right? You have the antigen that's going to be binding to the receptor. You're going to start to form colonies. So they're going to start to develop more and more of them. Some of them will become memory B cells, which are then going to be utilized upon subsequent exposure. And they're going to build faster and harder than before. And then some of these down here are going to differentiate from plasma cells into the antibodies, which then again could cause the foreign cell to die. Okay, so antibodies. These structures here are called immunoglobulins. They are going to be Y-shaped molecules that have two identical long polypeptide chains. And then they also, and that would be, let me circle them. Oops, let me circle them for you. So the two long chains are going to be the middle two, that one and that one. And then you have the two short chains on the outside, there and there. And those are going to be light or L chains. Uh, the antibodies or the immunoglobulins are going to be a constant portion that binds to the complement and then phagocytizes. The variable portion And again, the constant portion is going to be the parts here, here, and here. The variable portion is going to be the purple parts at the ends. Is going to create a unique shape that allows recognition and binding to the antigens. So what do they do? How do they function? Antibodies are going to be for recognition and binding to the antigens. They are going to be in charge of inactivation of the antigen, and they're going to form the antigen antibody complex, which is going to be present in order to neutralize the toxins and prevent viral attachment to the cell membrane. This also causes a loss of mobility in some of the bacteria, which is a good thing because then they can't move. And this is a part of the complement activation system. And again, it works as opsonization. When talking about cell-mediated immunity, you'll have defense against the infected cells, parasites, fungus, protozoans, and cancer. And the T lymphocytes are going to mature in the thymus. You'll have two types that are going to mediate cellular immunity. Those are going to be the cytotoxic T cells and then the helper T cells. You're going to require presentation, which literally just means like, I'm bringing this foreign thing to you of the antigen to develop clonal expansion. And then after the infection is gone, some of them again are going to become memory and others will just die. Okay, so that takes us to the major histocompatibility complex 2. So class 1 antigens or MHC1 are going to be molecules that are built into cell membranes. Here you're going to then have the antigen presenting cells that also display MHC2's antigens. And those are going to be like the macrophages, the B cells, the thymus cells, and then the dendritic cells. So you'll have the antigen presenting cells, like macrophages and B cells, that are going to ingest or engulf the foreign particle and then display them as a part of the MHC2. So this would allude to the fact that the antigen has been presented to the T cells. Okay, and then what do the helper T and the cytotoxic T cells do? Well, an antigen MHC2 complex, along with what we call interleukins, are going to stimulate mature helper T cells. They're going to activate them and undergo clonal expansion. And then those helper T cells are going to have an effect on both the cell-mediated and antibody-mediated immunities. So it kind of looks like this, right? This yellow part over here is going to be the antigen-presenting immune cell. You're going to have the MHC2 here that binds to a receptor, which we call a CD4. And then from there, it's going to bind to the helper T cell. Then in response to the cytokines, your T cells are going to start to clone themselves, which you can see here. And they're going to then produce different cytokines that are going to activate the B cells, like here, that are going to bind to CD8 receptors and become cytotoxic T cells. Those cytotoxic T cells then have the ability to kill the foreign object, just like this. Okay, so helper T cells and B cells. So your activated helper T cells are going to secrete what we call interleukin-2s. This hormone stimulates the B cells, the cytotoxic T cells, and the natural killer cells. The B cells are going to become activated plasma cells. And then from there, the antibody-mediated immunity is going to be regulated by the cell-mediated immunity or helper T cells. Then you'll activate the cytotoxic T cells, and they will interact with MHC1 on the infected cell and destroy it. How they destroy it is they create these perforins. A perforin is literally like a perforation, which is a hole in the cell membrane, and it acts kind of like a MAC, where you're opening up a hole for water to rush in, the cell to swell, and then eventually lyse. And this is kind of the same idea here with the immune system interactions and how there's a lot of different components. I'll leave this one to you guys to follow through because that was everything I just said. Okay, and then activation of the immune response. You have antigen detection, activation of the helper T cells, antibody production by B cells, and then actively creating specific antibodies and memory cells. So we'll start off with induced immunity. This is where you have active and passive. Active, again, is going to be the production of antibodies against the antigen. So it's going to be infection, which then causes memory B cells to activate and then T cells to activate. could be due to immunizations, and the long-term response is what we would call a secondary response. So you guys will note here, when first exposed, antibody concentration is relatively low, right? It only gets up to about 1 unit per milliliter. Upon second exposure to that same pathogen, look at how fast and how high the antibody concentration goes. It goes up to 100% in 10 units per milliliter, which is crazy fast, right? This, when you're looking at the units, because it has a longer graph, is the same as it takes to get to the peak. So here you're about a 10-ish. Here you're about a 10-ish. So you're less than one here, and you're over 100 here. That's crazy big difference. So basically what we're saying is that your memory cells are going to help produce a stronger and faster response. And then the last part here is passive. This is where you have uh, the prepared antibodies, like you'll have them take place at the placental transfer between mom and the fetus, in breast milk, and then in gamma globulin injections. These ones are going to be more short term. Okay, so that takes us to the end in our clinicals. Homeostatic interactions with your immune system. Like your integument system is part of your innate immune system, which means that the skin is it's like a first line of defense to protect anything from getting in. Your skeletal system has bone marrow, which is the site for B and T cell production. Your muscular system helps move the lymph drainage back to um, like the thoracic duct. The nervous system has immune system responses to help protect the brain and also inflammation. The endocrine system is going to allow lymph to help enter the bloodstream. I'm sorry, help blood deliver endocrine hormones. So that's where the hormones are too big that they'll move through the lymphatic vessels. Then you have the cardiovascular, which again, lymphatic fluid returns excess fluid created in the capillary exchange into your venous system of your blood. Respiratory system, like the PCCE that lines the trachea. Urinary system, like malt and the acid properties of urine. The digestive system, which has the tonsils, the malts, the stomach, and they all protect the GI tract against pathogens. And then again, the malt and acid production in like the urethra of the males or the vagina of the females to help protect them. Okay, clinical issues. This is called lymphedema, or short, it's called edema. Basically, it's your body's inability to return excess extracellular fluid to the venous system. In this case, it really just pulls towards your lower extremities. And because the fluid can't move up, it's going to start swelling in the lower extremities. This could lead to serious complications like lack of blood flow, which could eventually cause like necrosis of the tissue and things like that. Lack of nervous supply, which means you can't feel the area anymore. So this is all lymphedema. This is of the hands. This is of the leg. And then the last part here, the immune system. So AIDS, you guys have heard of, is acquired immunodeficiency syndrome. This is where viruses are going to help attack your helper T cells and render them ineffective, which means your body can't heal very well on its own. It makes you susceptible to getting sick very often. Also, allergies. Allergies release histamine by the mast cells, which results in inflammatory responses. And then autoimmune diseases are where your body has a hard time differentiating self from non-self cells, and accidentally it kind of depicts or lets your body know that your body's own self cells are foreign and it attacks, unfortunately. So like lupus or systemic lupus is an autoimmune disease, rheumatoid arthritis, type 1 diabetes, right? Those are all where your body is attacking itself, unfortunately, and killing off stuff that is beneficial. Okay, you guys, that ends it for today. If you have any questions, let me know.